visiting PhD student, actually from Germany, but she's doing her PhD with Pablo Buraco and myself. So she's here for 5 months in total, and she will present her PhD project as a normal PhD plan, as usual here at EBD. Colette, the stage is yours.
So thank you all for coming along today. So today I'll be discussing, as she mentioned, my PhD project, which is focusing on a multi-level perspective on the effects of microplastics on amphibians. But before I do so, I'll give you a bit of background about myself. So I'm from Scotland, and I did my bachelor's and my master's at the University of Glasgow. And during my bachelor's, I did my thesis on the effect of chemical pollutants on amphibians, alongside Pablo Buraco and Pat Monaghan. For my master's thesis, I continued the theme of ecotoxicology, and I looked at the uptake of microplastics in freshwater birds in the environment alongside Pablo Salmon and Sophie Sfaris. After my master's, I came here for 3 months to do an internship, and during this time, we looked at the effects of microplastics on amphibians, and now I'm based in Germany in the Technical University of Braunschweig. And my PhD is 3 years long, and it's funded by the German Research Foundation. And for this project, I've joined the lab of Katharina Ruthsatz, and my co-supervisor is Pablo Buraco. And we have a range of collaborators throughout Germany. So the reason that we've decided to focus on amphibians is because 41% of species are at risk of extinction, and they are the most threatened vertebrates. And this is due to a range of different factors, including pollutants. So this includes chemical pollutants, which include agricultural pesticides and herbicides or pharmaceuticals, for example. And in the environment, they are referred to as a cocktail of pollutants because amphibians are not only exposed to a single type of pollutant, but multiple, and this can intensify their toxic effects. There's also plastic pollutants, and my particular focus is on microplastics. And these are plastic particles within a size range of 1 micrometer to 5 millimeters. So there are 2 types of microplastics. There are primary microplastics, and these are plastics that have been manufactured to be within this size range. So this includes exfoliants, for example. So the microbeads in exfoliants are manufactured to be within this size range. But you can also find secondary microplastics in the environment, and these are when larger pieces of plastic are broken down through biological, chemical, or physical processes. So as I mentioned, they come in a range of different sizes, but they also range in their polymer type and their shape depending on where they were sourced from. And they come from a variety of different sources. So freshwater river ecosystems have multiple entry points of microplastics. This includes urban runoff, industrial waste, landfall waste, agricultural sewage sludge. And also, because of their light density, they're able to be carried up into the atmosphere and they fall back down through rainfall. So they really are found really widely distributed throughout the environment. And inland, they are more abundant because, as we can see, there are multiple entry points into river ecosystems, and as they enter the coastal regions, they tend to dilute out a bit. And unfortunately, they're highly prevalent in amphibian breeding ponds. So it's been confirmed that microplastics are being taken up by amphibians, and they are particularly vulnerable because of their permeable skin and their unselective feeding behaviour, and also their limited dispersal capacity. So during their early life stages, if they are semi-aquatic, they are restricted to freshwater habitats until they metamorphosize and develop legs and have the capacity to evade these habitats. And they have also been found to accumulate in some organisms, in some amphibians. And we are also interested in looking at cross life stage retention. So do they retain them throughout their different life stages as well? And a novel part of microplastic research is the focus on biofilm. So biofilm is a layer on the microplastic particles consisting of different bacteria, fungi and algae, which can create its own environment. And the likelihood and the types of organisms growing on the microplastics are dependent on various different factors, including the shape and the size of the microplastic, surface roughness. So there are multiple different factors that contribute to whether or not they'll grow. So the first chapter of my PhD was a meta-analysis where I focused on the effect of chemical pollution on oxidative stress in amphibians. For my second chapter, I looked at the uptake of microplastics in filter feeding larvae. For my third chapter, We really wanted to look closely at biofilm formation on microplastics, in particular the microplastics that we were using in our own experiments. And for our fourth chapter, we wanted to create a more realistic view on biofilm colonized microplastics, and we fed these to the amphibians. So as I mentioned, my first chapter was a meta-analysis, and for this we looked at the effects of chemical pollution on oxidative stress in amphibians, and this work was published last year in Proceedings B. So the aims of this project were to look at a variety of different chemical pollutants and look at how it's affecting oxidative stress and oxidative damage at different life stages, and for this we included the embryonic life stage, the larval life stage, and the adult life stage. And for each paper to be included in our meta-analysis, they had to meet each one of these criteria. So firstly, they had to have, for both the control and the treatment groups, the oxidative stress values, or the mean oxidative stress values, either the standard deviation or standard error, and the sample size. They also had to specify which developmental stage they studied, and they had to only test with one pollutant. So if there was a study that included 2 pollutants in combination, we did not include those values. And finally, they had to include at least one biomarker of oxidative stress or oxidative damage. So overall, we included 104 studies in our paper, and we had just over 2,000 effect sizes, and we included 35 species. And these are the results looking at the effects of oxidative stress across the different life stages. So so these points on the graph are showing the effect sizes, and in blue we have the non-enzymatic responses. In the orange we have the enzymatic responses, and in yellow this shows whether or not there was oxidative damage. And on the top left-hand corner we have the effect on the embryo life stage. So here we can see that there was a low effect on redox machinery, whereas in the tadpoles, which is on the bottom left-hand side, we did observe a slight increase in the non-enzymatic and enzymatic parameters, and we did not observe an effect on oxidative damage. However, in the adults, we saw a conversing effect. So we saw a weak increase in the levels of the enzymatic and non-enzymatic parameters, but we did observe a large increase in oxidative damage. So what we've observed is that in the early life stages, they appear to be increasing their antioxidant response, which in turn reduces oxidative damage, whereas in the adults, they haven't had the same increase in their antioxidant response, which in turn can cause oxidative damage later in their life. And during our project, during this investigation, we also realized that there was a gap in the study on microplastics and redox machinery. So this is what we have taken forward and studied in chapter 2. So for our second chapter, we are looking at the uptake of polyethylene microplastics in filter feeding tadpoles, and we're looking at their effect on growth, oxidative stress, telomere length, and corticosterone. And we used the African clawed frog, which is an aquatic species, and they are filter feeding. So this is ideal because they are non-specific in what they ingest, so they are more likely to ingest microplastics in our experiment. So for our experimental setup, we had the control group. We had one group with the polyethylene microplastics and one with cellulose fibers. And we also had a group where corticosterone was either present or absent, and all of the buckets were kept at 24 degrees. And we added exogenously, and corticosterone is a stress hormone that's released by the HPI axis, and under stressful environments it's released, and it can accelerate the developmental rate of the amphibians. So we also used polyethylene microplastics because this is a relevant pollutant type. It's found in the amphibian breeding ponds. It's the most prominently found plastic type in the environment. And the concentration we used is also economically relevant, environmentally relevant, and it stimulates a scenario where there's a higher level of pollution. And we use cellulose fibers because they are within the same size range as the microplastics, and they are naturally found from plant sources in the environment. So for the corticosterone assay, we used the tail, and for the oxidative stress, we used the head. And for telomere length, we were particularly interested in the liver and the gut because the liver is the site of detoxification and metabolism of foreign substances, in this case microplastics. And the gut is particularly interesting with microplastic research because it's the first point of contact with the microplastic and the tadpole. And in an event where there's maybe inflammation, this can increase oxidative stress and oxidative damage, and in turn cause telomere shortening. So here, looking at this graph, on the y-axis we have the body mass, and on the particles we have the fiber group, and these are the cellulose fibers, and we have the microplastic group. And then on the green bars, you can see where QORT was absent, and in the kind of orange beige graphs, these are where QORT was present. And if we look at the graph here, the microplastic treatment reduced the body mass by around 18%. And on the other side, if we look at the effect of QORT without any of the particles, it reduced the larval body mass by 16%, which is expected because, as we can predict, a release of QORT will accelerate development and reduce the body mass. However, we did not observe an effect of microplastics on the QORT levels. And for the next few graphs, I'm only going to focus on the left-hand side of the graph because we didn't find a significant interaction between the presence of QORT and the plastic and non-plastic treatments. So looking at the left-hand side of the graph, we can see that there was no significant effect of microplastics or control fibers on the QORT levels. And similarly, we also found no effect on oxidative stress or oxidative damage. So here we can see, I've just used one of our enzymatic indicators of oxidative stress, but they all showed the same trend. And on the right-hand side, we can see that there was a slight increase in oxidative damage, although this was not significant. And similarly, we found no effect on the telomere length in either the gut or the liver. So overall, what we found was that there was a reduction in larval growth, but this did not have any carryover effects on oxidative stress, QORT, or telomere length. And one explanation for the reduction in the larval body mass could potentially be because of nutrient density. So under the assumption that they ingest the same amount of having one treatment where there's microplastics present, reduce the chances of taking up the algae. So this may have reduced the nutrient density in our treatments. So we were interested in observing why we didn't observe any effect on QORT or telomere length, and that's going to take us on to chapter 4. But before we move on to chapter 4, we were interested in looking closely at biofilm formation and which factors can increase the growth on the microplastics that we're using in our own experiments. So this takes us on to chapter 3. And just to give you a quick explanation of how biofilm forms, they first of all, so this blue circle is meant to be the microplastic, and they adhere to, or they attach to the microplastic, and they form this extracellular polymeric substance which allows them to stay onto the microplastic, and they proliferate, and they can either break off and disperse, or they can be ingested and carried through into the organism when they ingest the microplastics. So the aims of our project were to look at how biofilm colonization on our polyethylene microplastics are influenced by environmental temperature, sampling time, and also particle type and size. So for our experiment, we used two different temperatures, 23 degrees and 28 degrees, and we had microplastics. We used the same microplastics, so polyethylene. However, this time, instead of using cellulose, we used ground mussel shell because this is also environmentally irrelevant. Mussel shell can be found in the environment, and they can be ground down to the same size range as the microplastics. And then we had another treatment with the control, and there were no particles present in this. And we had two different sizes, as I mentioned, so small and large. And we collected pond water, and this pond water was added to our flasks containing the microplastics particles and the mussel shell particles. And then we waited until 15 days, and we sampled half of the samples, and then after 31 days we sampled the rest of them. And to try and look at what was growing on the particles, we decided to filter the water and basically cut out a part of the filter paper that had the particles attached to it. And then we extracted the DNA from this piece of filter paper, and we used water swabs because we were also interested not only what was growing on the particles, but also if there was any differences in what was growing in the water as well. So we extracted the DNA and purified it, and we sent it to be sequenced. So before I came here, I finished this, the lab work, so I've yet to properly analyse the data, but we do have some preliminary data that I can show. And so what we can see so far is on the y-axis we have the alpha diversity, and this is the mean species diversity. And on the bottom slide, we can see the different particle types and particle sizes. So mussel shell and our plastic particle. And then we have the no particle control, and this is our flasks that contained only pond water. And what we can see is that the small and large microplastic particles do appear to have a higher alpha diversity in comparison to the control. However, we observed no effect of temperature or sampling time on the alpha diversity. And these graphs combined the data from all of the different treatment types. And again, these results are preliminary, but so far we can see no difference in the alpha diversity. And we also have some really impressive photos of, that have been provided to us by one of our collaborators, Matthias Munsken, and he has access to a scanning electron microscope machine. And he's been able to look really closely at the plastic particles themselves and to observe if there's any growth. So in this photo here, we have a pristine microplastic. I took some autoclaved water and I added the microplastic particles and filtered it. And so this is a pristine microplastic without any modifications. And then we have this large microplastic here, and there does appear to be some sort of maybe something attaching to them. We still need to look at this closely to see what's attaching to the microplastics, but it's incredibly detailed, and we can see already that there does appear to be something attaching. So the next steps are, of course, to properly analyse the data, and also to look at beta diversity, to see which taxons specifically are growing on the microplastic particles and the mussel shell particles. And that takes us on to chapter 4. So this is the work that I'm currently conducting here at the EBD. And we are aiming to look at how biofilm colonized microplastics affect the gut health, and we're looking at a multi-level effect, and how it affects organ accumulation in the amphibians. So we're using the western spadefoot toad, and this is different from our previous study species, which was Xenopus, and this is because these are semi-aquatic and they have extremely long developmental periods, which makes them ideal for plastic research because it extends the period of time that they're exposed to the microplastics. And they are also grazers, so our previous study species were filter feeding, and these are grazers. So this model here, we're looking at the lining of the gut, and so far what we're interested in is the gut, because again it's the point of contact first with the microplastics as they're ingested, and what we want to know and understand is, are they causing inflammation or damage to the gut lining, which we're going to look at using histopathology analyses, and also whether any potential inflammation can have negative effects on the gut microbiome and oxidative stress, in CORT, for example. And secondly, we're also interested in whether the microplastics can pass through the lining of the gut and be carried through the blood system into other organs in the body. So, and also, finally, if they can retain the microplastics across different life stages. So here are some samples that I've been working on here at the EBD. So our first sampling stage was Gosner stage 35 to 37, when they're still in their larval stage. And as you can see, and our second stage is once they've metamorphosized, we are sampling at stage 46. And as you can see, the gut undergoes extreme remodeling during this period. So in the larval stage, it's much more coiled and long, whereas when they reach their juvenile stage, it's much shorter. So our experimental design includes 2 different particle types. So again, the polyethylene microplastics and ground muscle shell particles, and we're only using a single size range, so these are between 34 to 50 micrometers. We have virgin particles and particles that we've aged with pond water, so we aged them in pond water for a week before adding this to the buckets, and we leave them in the buckets for a week before cleaning the buckets and restarting the process. And we're looking at, again, the multi-level effects of microplastic ingestion on amphibians. So we're looking at the organism, organ, cellular, and molecular level. So we want to understand how it's influencing things, for example, growth, tissue accumulation, inflammation, and we're going to be looking at the metabolomic profile as well, and gut microbiome and indicators of stress. And for the second part of our project, again, we're going to be looking at the accumulation in different organs, and also whether it's continuing to different life stages. And the organs that we have chosen to look at include the lungs, the heart, the liver, the gut, the brain, and we're taking tissue from either the tail or the hind limb. And then we're using fluorescence microscope to look at whether they're accumulating in the different organs. So although I don't have any preliminary results yet, we can show that we're able to confirm that they're definitely ingesting the microplastics, because as I mentioned, these are grazing tadpoles, and we can observe them grazing the microplastics on the sides of the buckets. And on the right hand side, we can see that they are releasing the microplastics through their feces, which confirms that they are definitely ingesting and releasing the microplastics. So the next steps are, of course, once I'm finished with my experiment here, I'll return to Germany and I'll be processing the samples for histology, metabolomics, the gut microbiome, and also the organ accumulation analyses. And this photo here isn't related to this particular project, but is an example of one of our collaborators in Germany, in Hamburg. She's provided this photo of the gut of a Xenopus tadpole, and this tadpole was fed microplastics. So these are the types of images that we'll be looking at for the tadpoles that we've measured here. And so overall, just to look at the progress of my PhD so far, Chapter 1 is complete and our work from this project has been published. For Chapter 2, it's basically complete, but we have a paper that's currently under review with minor revisions in the Journal of Experimental Biology Part A. And for Chapter 3, I've yet to do the data analysis and the write-up. And for Chapter 4, I'm still doing the live experiment here, and once this is complete, I'll process the samples, conduct the data analyses, and then I'll complete the write-up. So firstly, I want to thank all of the people here at the EBD and my collaborators in Germany, and of course I want to thank you all for listening to my talk today. Thank you.
Thank you very much, Colette. Now we have time for questions.
Well, fantastic. I mean, obviously it's doing great stuff. I'm just wondering if the ingestion of the microplastic particles is reducing growth because basically the kind of satiation effect that it's replacing useful food in the gut, so it's actually reducing the total ingestion rate of food, right? That's what you were suggesting. Could you not estimate if it was the pure effect of that? Would you not be able to estimate somehow from what's already known with the physiology and metabolics of just how much growth effect that should have? You know, to look whether there's kind of an additive effect to the fact that it's a plastic rather than some other inert particle. I'm just wondering what you think about that. And also the last chapter, in the chapter 3, I mean, I think about other aquatic organisms as well. Presumably you could do the same kind of experiments with other models, like invertebrate models as well, once you know that there's a biofilm there, and look at how that might affect, I don't know, something like a filter feeding brine shrimp or something in terms of ingesting those kind of things. So any future plans to do any work on any other organisms to see other effects?
Yes, so in response to the first question, I think that's what we're interested in investigating further. It's to look at these additional molecular mechanisms that might have kind of affect why they were reducing their growth. So in our current project in chapter 4, we'll be looking at the metabolomics, so the pathways, and also the gut microbiome. So we're going to be looking closely at the gut and why this, so like looking at a further explanation, because this first theory was just a hypothesis, but we're going to look more closely at answering the questions, I think, if that answers or makes sense. But and for the second point, currently, no, we're just focusing on the amphibian species, because these are extremely useful models because they're so plastic as well, so they respond extremely well to different changing environments, especially during our early life stages. So currently there's no plans to continue with another species or model organism, but I think once we understand the mechanisms driving the effects on the tadpoles, you can apply it to other model organisms and test the effect of biofilm growth.
Okay, thanks. Yeah, just to be clear, I was just thinking, if you can quantify that, let's say 10% of what's ingested are plastic particles, so your ingestion of food is reduced by 10%, then can you then make a theoretical prediction of just how much that affects growth? You know?
I see what you mean, yeah. That could be definitely an approach that you can take, that's true.
Yeah.
To maybe answer that question, if it is, because we did assume that they were ingesting the same amount of particles, so that would definitely answer that question and do like a percentage-based approach, but yeah.
Do we have more questions? Otherwise, we have one online. Paula Hidalgo says, thank you very much for your talk. Maybe I missed this nuance, but apart from ingesting the plastic, is it possible that they also get microplastic inside the body through their permeable skin? Thanks.
Yeah, that's true. That's what makes them particularly vulnerable to microplastics or stressors in general, is because of their permeable skin. So maybe, I guess we'd have to investigate the size range and how this maybe affects the uptake of the microplastics, possible route, and through their gills as well. So that's something that could also influence how they're taking up the microplastics and what makes them particularly vulnerable. Yeah.
Yeah, thank you very much. So have you considered perhaps a more evolutionary view to your thesis in terms of how plastic density is affecting lifespan, life expectancy, the quality of the progeny, population size, these kind of things?
Yeah, I think for that type of approach, you'd need a much longer experimental period. So that's definitely an approach you can take, because that's something that's extremely worth looking into. So how it carries over to, because we're currently ending the project, it goes from stage 46, but it's also important to understand how it affects the later life stages into adulthood as well. So that's definitely an important approach to take as well. It's not something we have the time to do for this particular project, but it's a really valuable insight into the effect of microplastic pollutants. But yeah, I don't know if that answers.
Oh yeah, but perhaps you know, this is a question for your defense. You know?
Yeah.
So you know, you can see the effects of the microplastics, but what's the effect in terms of fitness for populations, right? Can animals and plants live with this new actor in the environment?
Of course, yeah, because we're looking at a molecular, or we're looking at different sort of levels of their effects. So from the whole body, so we're looking at whether it affects their growth, their developmental rates, their body mass, for example. So those are parts, or those are aspects we will be looking at. And we'll also be looking at the molecular level, so the metabolic pathways, whether it affects their oxidative stress, core, the gut microbiome. So we will be looking closely at the mechanics sort of driving any of the effects of microplastic pollutants. So yeah, I think we'll be taking that approach, just not into adulthood, but that's a really important part.
Your thesis is halfway, you know, plenty of time till 2027, you know, to add something else.
That's right. Thank you.
Do we have more questions? Is there one online more? Okay. there aren't any more questions. Thank you very much. Also, thank you again, Colette.
Thanks.
And see you all next week.